Welcome to the Soil CRC webinar today. My name is Michael Crawford. I'm the Chief Executive Officer of the Soil CRC. I'd like to begin by acknowledging the traditional custodians of land on which we meet today, wherever you may be in Australia or internationally, and I'll pay my respects to the elders past and present. I extend that respect to the Aboriginal and Torres Strait Islander peoples watching on this webinar today. And I extend a very broad welcome to everybody who's attending today. This is our last webinar in our series this year, before taking a break over Christmas in January. It is also our largest attendance for this year, recognising the great interest in this topic around soil biology and some of the work of the Soil CRC. It comes on the back of our World Soil Day, 5th of December, last Saturday. And I hope you all recognised and celebrated accordingly, whatever that might be. Normally at this time I advertise future webinars, but we have no specific webinars planned, as I was just saying, until we come back in February. We'll resume the program. In the meantime though, I do encourage you to perhaps go back and look at our webinars that you might have missed. We have them all online on our website at soilcrc.com.au. And whilst you're there, remain notified about future upcoming webinars and other activities of the Soil CRC through our Twitter, through our LinkedIn, and you can even access the webinars directly through our YouTube channel. Today's webinar will be recorded, as all our webinars are, and you'll be able to therefore access it in your own good time after today. Also, today's webinar was initially advertised from 12 o'clock to 12:45 Australian Eastern Daylight Time. We extended it to the end of the hour, to the full 60 minutes, partly because of the large number of attendees that we've got, and we anticipate quite a bit of question and discussion time at the end, and partly because in Lukas's presentation, there's quite a bit of information he's going to cover, so we've extended it. If that means you can't stay to the end, don't worry, you can go back and watch the recording on our website later on. So, recognising a very broad audience today, both nationally and internationally, a few words about the Soil CRC, the Cooperative Research Centre for High Performance Soils. We were established in 2017, 3 years ago, with 10 years of funding from the Australian government and from our participants and partners. We have 40 participants in the Soil CRC, including 8 universities, 4 state government agencies, 8 industry groups, and importantly, 20 grower groups from across the country from all states. Collectively, we come together to undertake R&D related to soils to help give farmers the tools and knowledge they need to improve their soil management and in turn to improve their productivity and profitability. You can find a lot more information about our projects, our research, our programs on our website, soilcrc.com.au. Following today's webinar, you'll have the opportunity to ask questions. We'll do that by typing them into the Q&A box, which you can find generally at the bottom of your screen. The box will pop up and you can type it in there. There is a chat function. You can have chat, etc. in the chat function by typing it in. But the questions, please place them in the Q&A box, and I'll read them out, and Lukas will be able to answer them at the end. So, introducing Lukas. Lukas is a program leader in the Soil CRC. We have 4 programs. Lukas leads program 4, Integrated Soil Management Solutions. He's a senior principal research scientist with the New South Wales Department of Primary Industries, and he's also an adjunct professor at both Southern Cross University and at Griffith University. He completed his PhD in agricultural chemistry and soil science at the University of Sydney in 1995. And his research interests include carbon and nutrient cycling, soil function, ecotoxicology, and the impacts of agricultural chemicals, and the impacts of management practices and climate change on soil resilience. What makes him well placed to deliver on today's topic, soil biology under the microscope. Lukas, thank you.
Thank you very much, Michael, and welcome everyone to the presentation today. As Michael mentioned, there's a very large audience and I think also a very mixed audience. So hopefully today I will be able to deliver information to the broad range of people attending the presentation. So fingers crossed that my IT works. Hopefully everyone's got their fingers crossed for me. And I think we're away now. So I'd also like to, in particular, thank you, Michael, in particular acknowledge Mick Rose, my co-author for the presentation today. And you'll see that I've got special, and I'll call it special contributions because there are some embedded videos and there's a whole heap of people who have contributed significantly to my presentation today. So rather than me giving you an introduction to the webinar, I've outsourced this to Professor Terry Rose from Southern Cross University. So handing over to Terry, who's in the field. Terry, as a researcher, what value do you see in testing soil biology?
So if we can understand soil biology or how soil biology affects soil resilience and then what consequences that has for crop growth and overall farm performance, we can actually exploit the soil biology to try and A, improve soils, B, improve crops, and C, improve the overall farm and environmental sustainability.
Tell us a bit about some of the Soil CRC field trials that you're running and what you're hoping to expect to see with regards to changes in biology there.
Yep, so basically we're trying to maximise the diversity of microbes and other soil biology in some of our treatments. And we're trying to do that by altering plant diversity in those systems. So by increasing plant diversity, we're hoping we can change soil biology. So we've got treatments including, for example, summer cover cropping and winter cropping systems, so some intercropping and some undersowing of multiple species underneath a cash crop. The aim of that is that diversity drives microbial diversity, changes your soil biology, and
from Felicity Gilbert from the Wheatbelt NRM. So I've got a bit of a recording from our discussion. So Felicity, you've been very active in Western Australia organising the Talking Soil Health workshops. Is there an appetite among growers to better understand soil biology?
I really think that's growing. I think traditionally farmers have been focused on the sorts of things that they can link very clearly to fertility around soil chemistry and physical properties. And I think in physical and chemical, quite often the linkages are quite obvious about where the constraints are. They're relatively easy to manage, and they're relatively easy to mitigate. Expensive perhaps, but certainly when you compare it to biology, fertility takes on a whole new, a whole new perspective, because I'm not sure that anybody actually still fully understands exactly what linkages soil biology has to soil fertility and how they impact upon plants and various other qualities. I think that this work is certainly gaining momentum and certainly gaining interest, and certainly anecdotally we are seeing more and more farmers interested in wanting to know about how they can actually stimulate biology. But biology in the soil, is incredibly complex, and there are parts that are good, parts that are bad, and their interactions with each other, with the whole food chain beneath the soil is incredibly complex and difficult to understand.
Thank you, Felicity. I think my job's made easy. So here I would like to introduce the soil biology testing framework that we've been working on for some of the Soil CRC projects. And in particular, I would like to acknowledge Mick and Anders Claessens for helping out with the diagram. So we've been developing this framework for the assessment of soil biology, and essentially we're using the framework to assess the improvement in soils, as well as making agronomic decisions. On the left, you can see where it all starts, and that is with the microbial food source. So the food source is derived from products of photosynthesis, either as root exudates, leaf litter, or soil organic matter that is processed products of photosynthesis. Then we move to the top of the screen, and it's essentially who's there. And there are many new methodologies that have been made available in the last few years to make this assessment easier. While it can be valuable to know who is there, this really is only a part of the picture when we're assessing soil biology, as there is significant functional redundancy in soil, and I wanted to stress that. By this I mean that if one particular genus or species is affected by a particular practice or event, there's usually some other microorganism that steps up and completes that same function. And that takes us on to function. So we move one across to the right. Here we can assess the potential of a soil to perform a particular function. This may be the ability to nitrify ammonium in soil, or it might be the ability to degrade complex carbon substrates. This can be measured by techniques such as looking at functional genes or enzymes. And we can see a little diagram there of a gene that we can measure, or an enzyme performing its particular function. However, the potential to undertake a function does not always relate to what has actually happened in soil, particularly when this is integrated over time, where there may have been fluctuation in soil moisture content or temperature. So very, very climate driven. This takes us to the assessment of the actual function that has been performed, that is independent completely independent upon who is there. So after we understood who's there and what they can do, there's still the lingering question around what does it really all mean? And is the soil better or is it more resilient? And in that sort of resilience framework, we can see a bit of aggregation there, and we can see the microbes and the roots working together. And interestingly, what happens now is in that resilient soil, we're actually driving that food source. So when we started to develop that resilient soil, we're actually altering the way the food source is made available to the microorganisms, and then that whole cycle that we can see can start changing. And I'd also like to just talk about some of the data categories, because I think that's pretty important in the way that we frame some of the work. So we've got research data, and this data is often complex in nature and is used to test hypotheses, scientific hypotheses. For example, to determine whether a particular practice, for example, mixed species cover cropping that Terry spoke about, improves the ability of the soil to cycle carbon and supply nitrogen, or whether the soil becomes more resilient. The data is often used to help understand some of the mechanisms that are involved, and I'll give a few of these examples later in the presentation. Then you've got data that farmers collect to verify a practice change. So biology data can be very useful to verify whether a practice has changed the biology, for example, increase the ability to cycle carbon. This is an example of the soil uranium experiment, but there are other great examples, for example, the soil health cards that have been developed by Soil Care for a number of industries. Now, this data is not actually used to make any immediate agronomic decisions, just to track how your particular change in practice is coming along. And then you've got data to inform a decision. So this type of data is very common when you get your soil tested for chemical status, where the test report will usually come with a fertilizer recommendation. This type of data, though, is far less common when assessing soil biology, but there are some important examples that I'll present later. Now, this decision icon is used throughout the presentation to highlight data that falls into this category. Keep an eye out for that little icon, because I think that's where some of the important points are going to be made. So we're going to go on to assessing the food source. This is kind of where it all starts. Now, I won't spend too much time here describing the various bits of equipment that help us to describe the microbial food source. Total organic carbon is commonly analysed by LECO, but there are several wet chemistry measures that can also be used. We also have a look at the carbon in the various fractions in soil. For example, the particulate carbon is a carbon in fractions above 53 micrometers in size and is more biologically available than the mineral protected carbon that may occur in fractions smaller than 53 micrometers. Essentially, the smaller the fraction of carbon, the more it has been microbially processed and the less bioavailable that carbon is for other microorganisms. Mick Rose has also been putting a lot of effort into understanding the quality of root exudates, as these are key drivers for selecting and controlling the rhizosphere microbial community, which I'll discuss shortly. And here we go. So fresh products of photosynthesis are key food sources for soil microorganisms. On the right, you can see one of Mick's chromatograms of exudates from rice, with each peak representing a different low molecular weight compound. And these include amino acids, organic acids, sugars, sugar alcohols, and phenolics. There's a general understanding that the more complex exudate profiles and those containing compounds that are harder to break down would favour fungal growth, while the more simple sugars favour bacterial proliferation. This Rhizobox experiment, which I've shown in several other webinars that I've presented previously, is a great example where roots from corn, and they're the little plants grown in the middle, gravitate towards the legume and away from the grass. In that case, I think it was wheat that was being grown. And that's most likely due to the complex interaction within the rhizosphere between these exudates and the biology that follows. So incidentally, that system was not nitrogen limited, so it had nothing to do with the nitrogen fixation of the legume. So this type of data starts to justify our work around mixed species cover cropping, integration of legumes and brassicas, for example, into systems where there are grasses. This is the NMR, Nuclear Magnetic Resonance facility at Griffith University, that has been used by Professor Chen and his team to assess the quality of microbial food sources from different systems. For example, they've done some work comparing food sources from tropical grazing to sugarcane and banana leaf going into soil. By being able to characterize the properties of the carbon going into soil, for example, as I mentioned, the quality of the leaf litter and the carbon after it's been processed by the soil biology, we can start to understand the mechanisms associated with soil carbon storage and how the carbon properties relate to the soil microbiology. An example here might be the bacterial to fungal ratio. So this particular spectra is assessing the quality of dissolved organic fraction, in the soil, and it shows that about 44% of that carbon in that DOC is very readily bioavailable and will probably be used by the R. strategist bacteria. So assessing who's there. So I could actually spend hours here exploring some of the methodologies for assessing who's there, but I've narrowed it down to some of the key assessments that we would regularly do in our research. Here I'd like to hand over to Justine Cox for a moment.
Hi, I'm Justine Cox. I'm a soil scientist here at Wollongbar. The microbial biomass carbon test measures the weight of the carbon from the microorganisms in a soil sample. Mostly this is bacteria and fungi. Biomass carbon is useful as it measures the fraction of the total organic carbon that comes from microorganisms. It's a measure quick to respond to land management and the climate. It is often around 1 to 5% of the total organic carbon in the soil. Biomass carbon is measured on site by the fumigation extraction method. Essentially, a soil sample is split into two, where one sample is exposed to chloroform vapour, which breaks apart the microorganism's cells. This releases all of the cell carbon and other nutrients. Both soils are then extracted with potassium sulphate, and the amount of carbon is measured by a LECO analyzer. The difference is due to the microorganisms. The data is expressed as milligrams of carbon per gram of dry soil. We can use the data for comparing treatments in a replicated trial or for monitoring over time. Biomass carbon is a very complementary measure to microbial activity. The percentage of biomass carbon to total organic carbon can tell us about the decomposition rate of organic matter in the soil.
that the biomass carbon test is often used in conjunction with respiration data, and that is how much carbon dioxide is produced by the soil biology. And that is to develop an indice known as the metabolic quotient. For example, we have shown that soils from an orchard with high residues of copper fungicide have a higher metabolic quotient than soils that are not contaminated. So this needs to be considered carefully when we're assessing soil respiration. What does it actually mean? So we can relate to this same phenomena. So when I'm stressed, for example, I'll have an elevated heart rate and I'll start sweating. And microbes in soil are actually not that different. So essentially, when they're stressed, they've got lower carbon use efficiency, and they start to release more of that carbon food source as CO2. In a very mature biological system, there tends to be lower respiration, high carbon use efficiency, and essentially that's converting more of that photosynthate, so that new carbon that's being produced by plants into stored soil carbon and less into carbon dioxide. qPCR, or quantitative polymerase chain reaction, is a DNA method that quantitatively assesses the abundance of specific phylogenetic groups of microorganisms in soil. Here is an important example from the PredictB team at SARDI that offers a service to analyse some of the key disease organisms in soil. The test has been calibrated to a risk rating for particular crops and is one of the few soil biology tests that results in an agronomic decision. That decision could be related to which fungicide to use, for example, which cultivar to plant, or even which species of crop to grow, or indeed avoid. And here's the first example of this decision icon that I made. I'm handing over to Anders Claessens.
My name's Anders, and I'm a PhD student here at the Wollongbar Agricultural Institute. I research mycorrhizal symbiosis in crops. Mycorrhizal symbiosis is important because it has the potential to improve the productive capacity of our cropping systems and also further us towards our sustainability objectives. I take in roots from the field and I assess them using a combination of both molecular and morphological techniques. Mycorrhizal fungi are in constant communication and exchange with the roots of their host plants. Fungi provide soil-borne resources to the host plant, and in exchange, host plants provide benefits to the fungi, including the products of photosynthesis. To more fully understand mycorrhizal symbiosis in crops, we amplify the amount of DNA found in soil, we amplify the amount of DNA found in roots, and we compare them. Then we scrutinise the DNA found in roots to establish which fungi have made it to the root and form the symbiosis from those mycorrhizal fungi available in the soil reservoir. To support our molecular data, we bring samples into the lab and scrutinise them under the microscope. By assessing the amount of root colonised, we can establish a relationship between the amount of DNA detected and the extent and growth development of the organism inside the root. By combining these data, we get a clearer picture of how assessing mycorrhizal symbiosis in sugarcane and in other crops can further our understanding in mycology and support our agronomic objectives.
Thanks for showing us down the microscope there, Anders. That was great. So taking some of what Anders was saying with regards to the quantitative PCR data for mycorrhizal analysis in both the soil and the root. So this particular data was obtained from a sugarcane trial. Now I will go and try and get my laser pointer going. So let's just have a look at comparison A with comparison E. So comparison A would be a summer and winter fallow, whereas treatment E, which is in the light blue, is a mixed species summer cover crop followed by a mixed species winter crop. So what Anders did is he went and sampled this particular sugarcane soil from the different treatments, and the quantitative PCR data is split into these different groups, and here we can see the taxa that are detected by the PredictB team in these different groups. And I'd like to acknowledge the Predictor B team for working closely with Anders in this analysis. What's interesting then is we go to the root, and we have far less diversity of AMF or mycorrhizae in the root than we'd actually do as inocular in the soil. And what's quite interesting data here is that the fallow, so where we had no cover crop for a period of probably 7 or 8 months, had much lower colonization than the mixed species, both summer and winter cover crops. Another important factor with this data as well is that there's a very strong gate effect, which controls the symbiosis between the plant and the fungi. So I've put a small decision icon here, as this type of data may be related to Longfellow syndrome, and some decisions on cover cropping, for example, can be made to manage the effects of poor colonization. I'll get rid of my icon. Rightio. So the feeding and life strategies of nematodes are well documented, allowing indices to be generated, which reflects the changes in the structure and also the function of the soil biota, providing a quantitative assessment of soil health. So the key features of the quadrates are, so we look at quadrant 1, for example, we have a stressed and degraded system, and that could be indicative of a flogged paddock and acid affected soil, for example. Most agricultural soils would fit into quadrants 2 and 3, with the aim of moving closer to quadrant 3. So we tend to move towards systems that are less disturbed. So for example, if we're going from a traditional till system into a no-till system, we'd expect to move towards 3. Some of the work that Gwen Greelett, for example, is doing with some of the regenerative farming systems, we'll be assessing this type of metric, and we would expect that there will be a move into quadrant 3 or possibly even quadrant 4. And examples of quadrant 4 might be native forest with a complex carbon input. You've got fungal dominated soil biology and a moderate enrichment of the nematodes. Data for this index can be obtained by both morphological nematode community analysis, and that's essentially looking at the community down the microscope, or by qPCR techniques. There are also opportunities within the framework that we are developing to use co-occurrence networks, which I'll explain in a little bit more detail soon, of other soil biological indicators, and place this data into a soil health quadrant example that we have here for the nematodes. I'm handing over to Mick Rose.
The fungal to bacterial ratio in soil has been proposed as one potential measure of soil health. This indicator can be measured by using strong acid to break down the cell wall components of fungal and bacterial cells, which are made up of different polymers. The cell wall of fungi is comprised of chitin, which is degraded into glucosamine by strong acid, whereas the peptidoglycan in bacterial cell walls is broken down to glucosamine and another amino sugar called muramic acid. These amino sugars can be measured and quantified by gas or liquid chromatography. The ratio of fungi to bacteria will vary depending on many soil factors, including pH, plant cover, nitrogen fertilizer, moisture content, and tillage, to name just a few. It's therefore difficult to interpret as a one-off measure, but it may be useful if monitored over time.
Thank you, Mick. Then we start looking at some more high-end techniques such as DNA sequencing. So how soil microbes respond to fluctuations in biotic and abiotic factors, environmental stresses, change in practices, or addition of an amendments, for example, is still a fundamental question in microbial ecology. The application of high throughput sequencing technologies has opened up opportunities to address this question. So what we have here is work from a PhD student that visited Wollongbar a couple of years back, and he was investigating the role of organic amendments on the soil biology. Structure analysis shows that some amendments have a greater influence on the fungal community compared to the bacterial community. So just very, very quickly, CK is a control. That's where we've got no amendment. And we can see here that the fungal community is dominated by Ascomycota. Where we have a couple of amendments, in this case, it was biochar with fertilizer. we start to have a lot less dominance of the Ascomycota and some of these other microorganisms come into play. Similarly, when we have a look at the biological community, in the control, we're very much dominated by proteobacteria. Where we have the biochar with other organic amendments, we start to have a much more influence of other bacterial genera, such as acidobacteria. So exploring large data sets generated by high throughput DNA sequencing requires new mathematical approaches and people that are a lot smarter than I am to move beyond the basic description of who is there or how biodiverse is the system. Network analysis of significant taxon co-occurrence patterns can help us to decipher the structure of complex microbial communities and their relation to measured soil variables such as organic carbon storage, soil nitrogen, and even aggregation. We'll just explore this in a second, but here we can see the influence of goethite, which is an ion amendment, on the networks between bacteria and fungi and some of the soil properties. So the green dots are related to bacteria of different genera, purple to fungi, and the orange are the edaphic or climatic variables, so the soil properties, for example, total carbon or soil pH. And the red lines are positive links and the blue lines are negative links. Now, you'll often find me staring at these types of diagrams for many minutes trying to decipher them. But yeah, we don't have that much time here to explore this in too much detail. But what I wanted to show you, and I guess a key feature here, is that where we have the ion amendment, and we're doing some research right now in the Soil CRC, looking at how ion amendments improve soil carbon storage, we can see far more complex interrelationships between both the bacteria and the fungi and these soil variables, in particular related to carbon storage and also aggregation. So it's a very clear sign here that the ion amendments are promoting soil aggregation as well as promoting certain fungal functions. I'd like to acknowledge Zwieten for this data. So she is wrapping up her PhD at Zhejiang University, and she'll soon be looking for some postdoc opportunities. So I just wanted to put that plug in for Zwieten. So enough done on the who's there. I could have gone on for many more minutes on other techniques for assessing who's there, but in the need to continue moving, we're going to now move on to assessing the potential to complete a function. So for example, soil enzymes, which are these guys here, are central to ecosystem processes because they catalyze a very wide range of reactions in soils that have biogeochemical significance. Catalytic soil enzymes can exist internally on the surface of membranes of microbial cells. They can be excreted into soil solution, so they can be free in the solution, and they can also be complexed in the soil matrix, for example, in the clays or on microbial debris. The assessment of functional genes, for example, by shotgun sequencing, can also describe the potential of soils to partake in a particular biochemical pathway. The advantage of assessing potential to complete a function rather than who is there is that we don't know what each of the microbial genera are able to do. And it also eliminates a confounding influence of that functional redundancy that I mentioned earlier, whereby other microbes may step up to do a particular job. Enzyme activity is often closely related to important soil quality parameters such as organic matter, soil physical properties, microbial activity, or biomass carbon that Justine introduced earlier. Soil enzymes tend to respond more quickly to practice change compared to other soil health indicators such as total carbon content. I do need to stress though that there are still quite some levels of uncertainty around the meaning and application of the data. For example, if a soil has high activity, or for example, phosphatase enzyme, does it mean that there is lots of substrate present that microbes are chomping through and hence there's plentiful phosphorus for plant growth? Or on the flip side, does it mean that phosphorus and phosphorus containing organic substrate is limited and the microbes are overproducing the enzyme to compensate? I would imagine it's probably a bit of both, but we do need to take this type of data with a bit of a grain of salt as well. And as I mentioned before, there are a whole range of functions involved in carbon cycling, nitrogen cycling, phosphorus cycling, sulfur cycling, and the list actually goes on for quite a while. Here's an example of an enzyme function. So chitin is the second most abundant polysaccharide in nature after cellulose, and it's found in the exoskeleton of insects, fungi, yeast, and algae. And chitinases are often or commonly produced by soil bacteria, in particular to gain access to this quite complex source of carbon and nitrogen. And interestingly, chitinase activity has been linked to disease suppressive soils. So there's still a lot that we can learn around what the particular enzyme functions mean. This is going back to some of the data that Anders showed earlier. And again, this is a sugarcane soil with treatment A being the fallow and treatment E being both the summer and winter mixed species cover crop. And we're assessing these particular practices over time in this particular case. And what we're seeing is that in general, where we've got the plants growing, in particular that mixed species cover crop, we tend to have higher activity of chitinase. There's some indication that we've got improved phosphorus cycling, improved sulfur cycling, and improved carbon cycling. So we can start to understand some of the key factors that are going on in that particular soil. Here I'm showing some data from a visiting scientist who came to DPI a number of years ago, Flavio Fornasia. And this is some of his data that he showed where he was doing biochemical mapping, showing essentially that truffle production is related to soil that has high phosphatase enzyme activity. So what we can see here is the orchard, and each black spot is where they did the biogeochemical mapping. So lots and lots of samples were analysed. And this area here, sort of where the red markers are, it's essentially a heat map, and the red is related to alkaline phosphatase, and the blue is related to lower alkaline phosphatase activity. So what Flavio showed here was that where he had high alkaline phosphatase activity, he also had the co-occurrence of truffles. So just a word of caution again in interpreting the results. So the high activity could be due to other soil factors that are related to truffle production, and high phosphatase activity does not necessarily mean you will get truffles. So if you're looking for phosphatase activity in your wheat crop, you're probably not going to get truffles. This is another technique that we're commonly using. So it's also known as the biolog method, and it reflects the overall microbial community activity and characterizes microbial functional diversity without the need to analyze individual microorganisms. This simple method has been widely used in microbial ecology, and here we can see a list of substrates that we commonly use at Wollongbar. The substrates go from simple sugars that would result in the proliferation of bacteria, in particular the R strategist bacteria, some of the sugars like glucose or fructose, through to the more complex molecules that would dominate, that would be dominated in fungal in soils that have got high levels of fungi, for example, a pyrocatechol. And this technique is again quite simple. So you've got your soil in the bottom, you put your carbon substrate in, you have a look at the respiration, the amount of CO2 is produced, is then trapped in the indicator gel, which you can then measure for the amount of CO2 produced. So it's a pretty neat little technique. And here we're looking at the effects of a herbicide on respiration. And there was a clear indication that even at label rate application, there was a small decrease in respiration. So here we've got the respiration on the y-axis, and along the x-axis is the herbicide concentration. This is the label rate. So as the herbicide concentration increased, the respiration went down. What is interesting is the understanding of how the soil microbial community develops as a response to the herbicide. So here, that same soil, when provided with protocatechuric acid as a substrate in the biolog test, shows that the community has developed to break down more of this complex aromatic substrate. Essentially, exposure of the soil to herbicide has primed the microbial community to be able to utilize more of this complex carbon substrate. So here, as a herbicide concentration goes up, we've got more respiration coming from that particular fairly complex substrate. I'd like to hand over to Cameron Copeland now, who's going to give us a short description of glomalin in soil.
Hi, my name is Cameron Copeland. I'm a PhD student at Southern Cross University with the Soil CRC. I'm here at DPI Wollongbar looking at bioavailable nitrogen, which is measured by extracting proteins from organic matter in soils.
Proteins are first extracted from the soil using a citrate buffer under heat and pressure in an autoclave. The sample is then centrifuged to remove mineral matter, and the supernatant is pipetted into microwell plates. Proteins in the soil extract are then quantified using the biosynchronic acid assay. This produces a purple colour which can then be read on a spectrophotometer. Autoclavable citrate extractable nitrogen has been shown to be correlated with aggregate stability, soil water holding capacity, and bioavailable nitrogen.
Thank you, Cameron, and thank you, Mick. And here I'd just like to stress that these glomalin-like proteins are microbial products, and we're able to link these with aggregates and ultimately to infill measures of improvements in soil physical properties. Here we're measuring saturated hydraulic conductivity. Interestingly, a recent meta-analysis published in the literature has discovered that one of the key outcomes from mixed species cover crops is indeed improved saturated hydraulic conductivity. And essentially this is showing that the glomalin fits in here. It's in these sort of complex nitrogen, extractable nitrogen proteins that you're starting to develop these microaggregates, which then through fungal activity start developing the macroaggregates, which make an improved soil for improved soil physical properties in soil. Okay, so now we're moving on to assessing the function that has actually been performed. So up until now, we've just had a look at who's there and what the potential is. But we'd like to take that concept one step further in actually assessing the function that's performed. So is it improved aggregation? Is it improved nitrogen cycling? Is it improved carbon capture and storage in soil? One of the key functions that we're looking at is known as potentially mineralizable nitrogen. And here I've got a great big decision icon that's showing up. So this is some data that we've obtained from undertaking the PMN test from sugarcane soils in northern New South Wales. So the potentially mineralizable nitrogen is essentially linked to carbon content and quality, the amount of microbial activity that's present, which reflects then the cycling of that organic matter, and then the development of mineral nitrogen in soil. And what's really important is that potentially mineralizable nitrogen can actually make up a fair amount of the nitrogen stocks that are available to plants and also microorganisms in soil. So understanding soil nitrogen stocks prior to making a fertilizer decision may well be one of the key strategies in overcoming high nitrogen loss pathways. This data is from the assessment of nitrogen stocks to 1 metre in depth in the sugarcane soils in 3 catchments across New South Wales. By adding the potentially mineralizable nitrogen to the soil mineral end stores, we have revealed that nitrogen stocks ranged between under 50 kilograms of available nitrogen per hectare to over 350 kilos of available nitrogen per hectare. So the problem with traditional measurement of PMN, or potentially mineralizable nitrogen, is that the tests can take a minimum of 14 days. And in our particular case, where we're looking at a sugarcane crop over 2 years, getting data from a 300-day test is not that meaningful because it actually can't help you with that particular fertilizer decision. Thus, integrating data from the traditional PMN test into fertilizer decisions can be quite challenging. So here we've got just briefly the ammonium to 1 metre in depth at these different sites. So here we've got nitrogen content, and that's just a number of sites. So majority of sites, 16 of the sites, had about 25 kilos of ammonium. The majority of them had very little nitrate, sort of under 50 kilos of nitrate. but many of them had quite high stores of mineralizable nitrogen. And yeah, we're looking up to 200 units. So when we add all of these together, we can see here that some of the sites had between 50 and that particular site, 350 kilos of nitrogen. If your sugarcane crop's going to be exporting 200 kilos of nitrogen, for these sites here, you might go, do I actually need to add nitrogen fertilizer? So that's a type of agronomic decision that, that is possible with this type of data. So mid-infrared spectroscopy and the model that we're developing integrates key factors associated with soil properties such as organic carbon content and also carbon quality to predict the PMN. So rather than having to wait a minimum of 14 days or maybe 300 days for the test data, which is now way too late to make a call on fertilizer application rate, this overnight test can predict with a reasonable level of certainty what the soil PMN data is going to be. So it should be noted, however, that this calibration is likely to be fairly specific for the soil types that we've analysed and also the crop. So in this case, it's sugarcane, and it would need calibration for other systems. And I guess a key point here is a test is only as good as its calibration data. The assessment of the enzyme that performs the function of breaking down cellulose is known as cellulase. However, as mentioned previously, the detection of enzymes may not necessarily correspond to an action being performed in the soil, especially over time. That's a bit like the PMN test. We have developed an MIR, mid-infrared spectroscopy test, to evaluate the biological breakdown of cellulose, which is the most abundant polymer that is deposited in soil via root and leaf litter. So here you can see that this particular spectrum, 2,900 centimeters, corresponds to cellulose. And when mixed done it in soil, we were able to actually look at how much cellulose is actually remaining after we've spiked the soil. And again, Mix obtained a stunning calibration. But again, it's important to note that that this type of data coming from the MIR is a prediction and it's not actually a true measurement. Some other types of outcome type related biological measures are how much nitrogen has been fixed. So data shown here are a snapshot of work addressing soil constraints that impact nitrogen fixation. In this case, we're looking at faba bean. Here we ascertain that low soil pH in combination with aluminium toxicity impacted both the plant growth as well as biological nitrogen fixation, which was significantly alleviated through the surface application of lime. Terry Rose has also been very active in understanding the contribution of nitrogen fixation from legumes in cover cropping and intercrops, as well as understanding both nitrogen loss pathways, as well as where and when that particular nitrogen becomes available in the crop. And this is using some stable isotope techniques. Now, unfortunately, I don't have time to go into too many more of these functions, but we can also use stable isotopes to measure the interaction of amendments with the native soil organic matter. And stable isotopes can also be used, for example, to measure the degradation rate of organic amendments, and also where and when in particular these organic amendments might mineralize and the nitrogen becomes available for crop growth. Another example would be soil greenhouse gas emissions by both the static chamber method or the auto chamber methodology. So bringing everything together, I guess the ultimate question is, is our soil better? And can we answer that by having a look at soil resilience? So a resilient soil will be better aggregated, will have higher carbon use efficiency and lower loss of carbon as CO2 or as leached carbon. So due to its aggregation, it will be better able to store water and deliver microbial functions going into stress. Here I would like to further explain the concept of resistance and resilience. So 3 soils were provided by Lawrence DeBella from a Soil CRC trial under Terry Rose's project. And this was actually supplied into Mayran Rashti's project. This included soil from undisturbed native vegetation on Lawrence's farm, a traditional fallow before planting sugarcane, and also that mixed species cover crop that I've been speaking about. Here that's shown in green, and I just call it mix. So the soil was incubated and the rate of nitrification, which is one of the many biological processes monitored, was tested over time. The soil was exposed to drying and changes to the rate of nitrification were measured. So here, a resistant soil is a soil that can continue to perform that particular function going into stress. When the stress was relieved, here by wetting the soil, the resilience is defined as how close to the original biological function that the soil returns to. So pre-stress, the nitrification rate was around here. After stress, it was a little bit elevated. So what's clear from the data so far is the fallow soil is both less resistant and less resilient than the soil with a mixed species cover crop. And I think we'll be speaking and using these types of metrics a lot more going into the future. And the conclusion for my presentation is given by Felicity Gilbert. So thank you, Felicity.
I think that something that is becoming really quite accepted by a large number of farmers out there is the idea that soil health is dependent on the intersection of biology, physical properties and chemical, and that sweet spot where all three are working in harmony together is something that we all want to achieve to maximise the health of our soils. I think that concept is coming forward very strongly. We're really getting there. I think the difficulty is in understanding how to achieve it and how to measure it. That is where we've still got work to do.
Thank you, Felicity, and thank you everyone for your attention. And I will stop sharing my screen, Michael.
Thank you, Lucas. Thank you for that comprehensive overview of a whole range of aspects related to soil biology. And yeah, 5 or 6 minutes left for the question time. We'll see how we go. So type them into the Q&A box. We've got 3 questions there already. The first was from Greg Chapman. Lucas, can you see the questions?
I can.
Okay, so Greg asked, what proportion of microbiology nutrition derives from plant exudates versus dead plant material?
Yeah, so going through the cycle, the initial food source would be both exudate, so root exudates, and also leaf litter falling onto the soil. As it's processed by the soil microbiology, it becomes less available. So we start talking about dead plant matter there. The actual proportions of those probably depend very much on the system. So for example, if you've got a sugarcane farming system that is green harvesting, you might be putting on 15 or 20 tons of dry carbon, mostly cellulose, onto that soil profile. If you're in a forest situation, there's probably a lot less input of labile carbon, and that's why you get differences in, I guess, the bacterial to fungal ratio between comparing, say, sugarcane farming systems and forestry. But the exact percentages, I probably can't answer that.
All right, good. And I should also add that you have the ability, attendees have the ability to upvote various questions. So they've then just jumped to the top of the list, as this one has from Brad Deegans. Measures of soil resilience being developed as part of this program, or any of the other investigations that consider measures of soil microbial biomass, diversity and function, also looking at relationships with resilience?
Yeah, the simple answer there is absolutely. Some of the work I showed from Mayran and Mix project, it was one example of nitrification, but we've also been using a whole range of other soil metrics, including biomass, having a look at respiration, and also some of the gene sequencing. So we're actually looking at actual genes and species that are present during these stress factors. I think there's a lot more work to be done around understanding the metrics of resistance and resilience, and how we can put those metrics into that type of of output, for example, the quadrant output that I presented for the nematodes. And I think that's where we need to go.
Okay. There's a question here from Michael Gooden, which has now got 4 upvotes. How can we have a professor, Jim Prattley from Charles Sturt University, talking yesterday at the Australian Farming Institute conference, saying that it is basically impossible to successfully store long-term carbon in Australian soils and we should not bother? How do we come up with accurate science and promote a correct message? And Lucas, I might just actually have a go at that myself. I've been fairly involved in this whole discussion. And the caveat being, I didn't hear what I do know, Jim, but I didn't hear it. But I do understand that there is a conservatism from many in the science areas around just how much carbon we can store in Australian soils. Conversely, there's also plenty out there who are perhaps overestimating or overstating the ability of our soils to store carbon. And it reflects a number of things, and basically the nature of our soils, our soil types, our climates across great parts of Australian agricultural landscapes, not all, but some of it. But to answer the last bit, how do we come up with accurate science and promote the correct message? Well, the correct message is based on accurate science. And so what we are endeavouring to do and what we are doing more of is collecting the data, relating it back to farm practice, agricultural practice, various interventions, from a whole range of farming systems and soil types and climatic situations. And from that, trying to improve our evidence-based understanding of the opportunities for sequestering carbon. I'll move on with that one. I'll go to Bill's question. Bill asked Bill Grant, have you looked at how long soil respiration is suppressed for after herbicide application? Days, weeks, or months?
That's a great question, Bill, and that really depends on the nature of the herbicide. So, for example, glyphosate impacts on the shikimic acid pathway, which is prevalent in plants, is not prevalent in soil. So there's actually very, very little effect of glyphosate, for example, on soil respiration. When we have a look at herbicides like atrazine, some of those biochemical pathways are shared between both the plants and the soil microbes. And there are, because of those shared biochemical pathways, herbicide like atrazine can impact some of the soil biological properties. So it depends on both the nature of the chemical, the bioavailability of the chemical in soil. So how much clay is present, how much organic matter is present will impact how bioavailable that particular herbicide will be. And the effects can last from days to potentially months, I would imagine.
Okay, thanks. Listen, given the time, I'll give people the option to log off in a minute and then perhaps follow up later. I'll give people the option for another minute or so to type in additional questions. We'll then draw a line and we'll answer these questions over the next 5 minutes or so. So we'll just go a bit longer, but we will bring an end to it shortly. So from Brad again, just jump to the top. Are we missing some of the functions and biomass of soil invertebrates in measures of soil biology, diversity and function?
So in my original talk that I was preparing, I had a whole stack of information on there on invertebrates, including earthworms and some of the tests that we use here for using earthworms. I didn't have the time to speak about those. So I focused the talk on bacteria, fungi, the mycorrhizae, and the nematodes. So apologies for that. But yes, that's an important point. It was ignored a little bit in this presentation, but it shouldn't be ignored in the broader assessment of soil health.
Thank you. From Ravjit Kangara, how much liquid carbon pathway contributes to soil organic carbon and how?
So
how much does a liquid carbon? Yeah, okay, yeah.
So I'm assuming that means dissolved organic carbon. So again, dissolved organic carbon tends to be that carbon fraction that is highly available to microorganisms. And for a microorganism to access most carbons, it needs to actually be in the liquefied form. It needs to be soluble. So again, depending on your carbon use efficiency in a stressed system, much of that DOC will go off as carbon dioxide. In a more mature biological system, you have improved carbon use efficiency, and more of that DOC might end up as long-term soil carbon via microbial processing. It'd be probably in the order of 1% or less, I would imagine. Maybe Mick Rose could confirm that number, but I would imagine that it will be a fairly small component.
Mick, are you coming on this one, or you're happy with Lucas's answer?
Yeah, happy with that.
Okay, good. All right. From Yusuf Faraz, does microbiology help plants to grow in sodic soils?
Yeah, and we're currently doing some of that work with Esan Tavakoli and some other colleagues right now. So we're certainly hoping that by alleviating certain constraints, they might be chemical constraints, we also start improving the biology and some of these biological functions that the soil can do, therefore improving the cycling of nutrients, and therefore getting that whole cycle going where we get improved crop production. So great question. Don't have an exact answer on that right now, but it's something that we've considered and that we're working on.
Thanks, Lucas. So we've got the last question now from Lewis Paran. How long does it take to achieve quorum sensing?
I'm going to hand over to Mick.
Yeah, thanks, Luco. Lucky last. Yeah, quorum sensing is one of these interesting phenomenon that bacteria and I think other organisms undertake. When a whole lot of them get together, they can sense that each other is there so they put out chemicals to basically tell each other that there's a sufficient quantity that they can start to change their phenotype and do special things. But it's not restricted to just plant growth promoting. It's not just restricted to beneficials, but pathogens also do it as well. So really it depends on the numbers of those organisms and if the conditions are right for their growth. So if the conditions are good for them to multiply, then they'll reach a threshold where they sense that there's enough of each other around them, and then they can start to do some tricky things like form biofilms and whatnot. So it really depends on the specific organism and the conditions and whether or not the conditions are right for them to proliferate. But yeah, quorum sensing is not specific to beneficials. So it's one of these interesting phenomena that, yeah, there's plenty of new research going on in that area.
Wonderful. So thanks, Mick, but very much thank you, Lucas, for that great overview and for handling the questions at the end. If people do have additional questions, please send that email to Lucas as per the last slide. But Lucas, just give us your email address again. Just say it out loud.
It's pretty easy to look me up on the internet, Michael.
All right. Okay. So thank you, everybody, and in turn, you can find it on our website. So I'll just take you back to that website. Where you can find that information. You'll be able to find this recording of this webinar in a few days' time in the under the webinar sections. I'll leave it there. Thank you, everyone, for your attendance. I wish you all a Merry Christmas and happy holidays over the period, and hope to see you all again in 2021 as we kick off our webinar program once again. Thank you, everyone.